vesicles and particles. Extracellular vesicles and particles are highly heterogeneous, ranging from the large apoptotic bodies to oncozones or bodies and oncozones to the small suprameres and exomeres and protein condensates. Newly discovered are the microsomes found in migrating cells, and exosomes are now distinguished based on large and small exosomes, and particles now include exomeres, suprameres, and protein condensates, as I just mentioned. So EVs can exert diverse functions in cells and tissues and organs by modulating many biological processes, from cell proliferation to metabolism to inflammation to angiogenesis. EVs are involved in transport and clearance of proteins and nucleic acids. Their cargo and composition are highly responsive to cellular changes. In medicine, EV can serve as diagnostic and prognostic biomarkers, as their cargo can reflect the cell origin and disease state. Most importantly, due to their low immunogenicity, EVs can be engineered for therapeutic uses. Okay, the myriads of functions are afforded by the complexity of EV composition due to the diversity of the encapsulated or surface-associated cargo, as we now call the corona. And these components include DNAs, RNAs, signaling proteins, and receptor ligands. But the effectiveness of these EVs relies heavily on the delivery of these EVs to the recipient cells. So this is an overview of the mechanisms of uptake from the most employed by cells, including caveolin, clathrin, and lipid raft-mediated pathways, to the cell-specific pathways such as macropinocytosis in neuronal cells, to phagocytosis in phagocytic cells, including macrophages. In addition, there are emerging novel pathways that are dependent on these membrane-associated proteins, as listed here, from sortilin, ARF6, RhoA, etc. So the use of knockdown and knockout technologies, along with the discovery of these specific inhibitors as shown here, allow us to characterize the pathways of interest. My goal for the talk today is to discuss these pathways to give you an appreciation for the diversity and complexity of EV uptake and cargo delivery. I just want you to know I made this slide on my flight here to Melbourne, so it's probably outdated by now, with all of these different inhibitors. And there's a new pathway of EV uptake that has yet to have an identified inhibitor. So let us start with the non-endocytic mechanism mediated through direct receptor engagement. Here's an example where EVs carrying the Notch signaling the Notch ligand delta-like-4, which activates Notch signaling, leading to downregulation of cell proliferation while increasing vesicle formation and branching. And this effect actually inhibits cell proliferation of those cells while enhancing the branching of the other cells, as shown here. So, along with non-endocytic mechanisms, there is the membrane fusion. And membrane fusion can be divided into snare-dependent and snare-independent. It is mediated by two transmembrane proteins, syntaxin and synaptobrevin, along with two other associated proteins, SN1 and SNAP25. This complex brings the two membranes in close proximity together, as shown here, allowing the lipids to mix to form what is called the Henle diaphragm. Next, the inner leaflet of the lipids then begin to mix to form the core, as you can see here. This mechanism is used by viruses to enter cells and is employed intracellularly for vesicle trafficking. And in a recent paper that was just published this year, these investigators further showed that an associated protein called MUC18C regulates membrane fusion by binding to this snare complex, allowing the vesicles to then bind to the snare complex and then mediate this internal membrane fusion. So there's also the snare-independent membrane fusion, and it is mediated through a protein called syntysin, and specifically proteins of the extracellular vesicle membranes are structurally rearranged. Their hydrophobic sequences are inserted into the target cell plasma membrane, which then undergoes lipid reorganization, protein restructuring, and membrane dimpling. In this way, it allows the plasma membrane of the vesicles to fuse with the plasma membrane of the cells of interest. So in a recent paper, it was shown by using a single EV trapped by optical tweezer, not only adhered to the surface of the cells, but also this single vesicle moved towards the cell soma and fused to the surface of this microglia cell. And then it disappeared from the surface and it dumped its content into the cell. Okay, now we're going to get into the endocytic pathways. And the first one is the caveolin-mediated endocytosis. This mechanism employed most often is mediated through these membrane structures called caveolae, formed by the integral membrane protein caveolin. There are three forms of caveolin. Caveolin 3 is found only in the muscle, so caveolin 1 and 2 form heteroligomeric complex in the ER. It then binds to cholesterol in the Golgi, and the complex is transported to the plasma membrane. This complex recruits other proteins such as cavins that phosphorylates caveolin, triggering this structural conformational changes to form a cup-like structure called caveolae. Next, the GTPase protein dynamin forms a helical complex around the neck of the invagination and it mediates the membrane pinching. This vesicle travels along the actin cytoskeleton to fuse with the endosomal compartment. Another endocytic pathway is the clathrin-mediated endocytosis. And what it is that this is the faster mean of uptake mediated through the interaction of EV membrane proteins on the plasma to the receptor or the adaptive proteins found on the plasma membrane of the cells. And this promotes the clustering of the soccer ball-like proteins called clathrin, which then forms a cage-like coat of approximately 60 to 120 nanometer in size. Again, membrane pinching is mediated through dynamin, and clathrin detached from the vesicles are now faded for sorting, recycling, and degradation. The third mechanism that is regulated through the endocytic pathway is the lipid raft, which are intensely packed, highly membrane, specialized microdomains, approximately 10 to 200 nanometer. They are extremely heterogeneous, highly dynamic, and they serve as the organizing centers for signaling molecules. These microdomains are enriched in sphingolipids and cholesterol. Planar lipid rafts internalization is mediated through a family of proteins called flotillins and other membrane proteins such as ARF6, RhoA, and GRAF-A. Macropinocytosis is a unique pathway of endocytosis characterized by the nonspecific internalization of a large amount of extracellular fluid, solutes, and membrane large endocytic vesicles known as macropinosomes. It involves actin and microtubule-dependent formation of membrane ruffles greater than 250 nanometer in size, and it's mediated by small non-rho GTPases such as CD42, RAC1, and 2. Fusion of macropinosomes to early endosomes require RAF4 and RAF5 and RAF24, while RAF7 mediates the movement into late endosomes. Further on, with the endocytic pathways, we have phagocytosis, and unlike macropinocytosis, phagocytosis is a receptor-mediated process through the distinct phagocytic targets typically recognized by phagocytic receptors. Following membrane receptor engagement, there is actin-dependent formation of membrane pseudopods called phagosomes, which are then fused with endosomes and shuttled to the late endosomes and eventually to lysosomes for degradation. Transcytosis is a form of specialized, highly regulated transport across the epithelial cells in the blood, in the blood-brain barrier mediated by clathrin, caveolin, and lipid raft-dependent endocytosis. This transcytosis allows EVs to cross the endothelial cells to get into the central nervous system and is released into the brain. Transcytosis may be derived from a similar mechanism used by pathogens to invade tissues. And finally, this is a newly discovered method, mean of endocytic pathway, and it's called the GLIC clathrin independent pathway. Some people refer to it as CG for short. It is a pinocytic pathway mediated by uncoated, so there is no coating, as you can see here. Tubulovesicular carrier called clathrin-independent carriers, CLICs. They mature into early endocytic compartments called GEECs, and they fuse then into endosomes through RAP5 and PI3 kinase. This pathway is used to import bulk fluid, folate receptors, as well as the following transmembrane proteins as listed here. So, we recently in our lab just reviewed studies that examine uptake pathways of 21 normal and cancer cell lines. 7 used a single pathway, 11 used 2 pathways, and 3 used 3 different pathways. The 3 most employed pathways are in order of popularity: caveolin-mediated endocytosis, clathrin-mediated endocytosis, and macropinocytosis. And although different cells appear to employ a dominant import pathway to receive EV-mediated messages, their ability to exploit multiple endocytic pathways to compensate for the failure of their primary import pathway ensures that this vital communication can still occur. So the knowledge that these cell-specific import pathways is one factor that we can consider when we are engineering therapeutic EVs to maximize their effective import into target cells. Now, EVs can maximize their effective import in the exploitation of multiple pathways of uptake ensures that this vital communication is never lost. But the limiting factor remains in the mechanism of EV cargo release. One mechanism is the direct fusion of the EV membrane to the plasma membrane, bypassing endosomal sorting to deliver the EV cargo. However, direct evidence of EV cargo release is minimal due to low sensitivity of detection. Mechanisms for endosomal escape have been proposed, including endosomal lysis, membrane proliferation, and fusion of EV into endosomal membranes. So today, we are excited to bring to you 4 exceptional talks First, Dr. Troyer will demonstrate that EVs engineered to carry retroviral envelope proteins enhances uptake. Then Dr. Twardowski will use engineered EVs loaded with transcription factors to target specific cells involved in intervertebral disc degeneration. Then Dr. Stewart will examine the role of phospholipid scrambling by enzyme scramblase in EV biogenesis and function. And finally, Dr. Hamadi will characterize EV crosstalk with tumor microenvironment using 3D tumor model systems. So with that, I would like to thank my collaborator, Peter Wormer, and my societies and funding agencies. Philadelphia is a rough and tough city to live and work in, but we do have the Love Statue, and of course, the Philly cheesesteak. So please come and visit us anytime. And to prevent you very smart people in this audience from humiliating me with rough questions, I'm going to go straight into the first talk, which is presented by Dr. Zach Troyer from Johns Hopkins University. And the title of his talk is Cellular Interaction and Uptake in Human Endogenous Retrovirus Envelope Displaying EVs. Thank you.
Hi. So I'd like to thank the organizers for giving me this chance to come and present my research today. I've been looking really forward to presenting this. So I'm Zach Troyer. I'm a postdoctoral fellow in Ken Whitwer's lab. And today I'm going to tell you about cellular interaction and uptake of human endogenous retrovirus envelope displaying EVs. So, sorry. Because my talk is very centrally associated with EV therapeutics, I wanted to first talk a little bit about some of the obstacles that are associated with EV therapeutics, some of which you heard about yesterday at the Education Day, if you were there. So this includes suboptimal targeting of desired cells, if you want your EV to go to this particular cell, and low levels of uptake by these cells. Additionally, if the EV does get to the cell, there's been shown to be low levels of endosomal escape and cargo release to the cytosol post-uptake. And lastly, and perhaps most importantly, there are immune responses in blood clearance of EVs. And specifically, Tom Driedomps from our lab had this paper where EVs were injected into a non-human primate model, and what was found is that with repeated administrations, EV half-life was reduced, and EV-specific IgG was increased, so it seemed like the EVs were being cleared from the blood. Now, a lot of these issues are related to the surface of the extracellular vesicles. So here I'm showing you a model of what typically happens when an EV is interacting with a cell and is taken up. So without surface modification, the majority of EVs taken up by cells end up being lysosomally degraded, and their cargoes are not actually delivered into the cell. So and there's plenty of literature out there supporting that this is the fate for most EVs. So how can we avoid lysosomal degradation? Well, a lot of research into EV therapeutics has found that if we can modify the surface of the EVs, we can alter their ability to bind to cells and fuse their membranes with cells to release their contents, where they can go on to have functional effects, as you see here. So what kind of proteins then can mediate this process? Well, a lot of people use viral proteins like VSVG to enhance the ability of EVs to deliver their contents. The pros of using such things, viral proteins, is that they're known to facilitate uptake and fusion in viral research. VSVG is commonly used with EVs even. Their receptors are well known, and these proteins readily get onto the EV surface. However, they have major drawbacks, including that they're very toxic to the producer cells, and they're highly immunogenic, which makes them not suitable for therapeutics. And if you think these proteins are the major targets of adaptive immune responses, which is why COVID vaccine uses the SARS-CoV-2 spike protein. So is there a way to harness the power of viral fusion proteins for therapeutic EVs without the drawbacks? We propose that we can use human endogenous retroviral envelope proteins. So what are HERVs, or human endogenous retroviruses? These are ancient remnants of retroviral insertions into the germline of primate ancestors. So basically, a long time ago, a retrovirus infected a germline cell, which was then passed down to offspring. So these proviral sequences can become endogenized in our DNA, and about 8% of our DNA is consisting of these sequences. Now, for most of these, they get mutated over time. And I wanted to show here just an example of the map of where some of these herbs are actually integrated, and you can see that they entered the human lineage at various points along the divergence from primates, from other primates, but we also share some of these herbs with other primates. But as I was saying before, most of these herbs end up being mutated. However, if there's a selective pressure or evolutionary pressure to preserve the function of one of these genes in this provirus, they can actually retain a coding sequence. And as you can see here, this is an example where a protein syncytin 1, which is one of these herbs, it's a viral fusion protein, and it's been preserved because it has a role in pregnancy and helping the placenta form. So given that many of these herb elements are viral fusion proteins, we asked, can they be placed onto extracellular vesicles? And so here's a little diagram of the 3 different herbs that I use on the surface of the EV. So the benefits of using herbs is that they're functional fusion proteins. They're present during early development, actually, and so we suspect they might be immunogenically invisible. And some of their receptors are well studied. However, in terms of cons, some do have unknown receptors. We don't know how well they can facilitate membrane fusion, especially on an EV, and we don't really know how well they're going to get onto EVs. And lastly, no one's really tested the immunogenicity aspect of this. So then we asked, can we get the herb envelopes onto EVs via overexpression? So just before I show you the data, I wanted to point out that these herb proteins typically have 2 subunits that need to be cleaved into a mature active form that can perform membrane fusion. So it's the SU and the TM subunits. Now, I collected EVs from cells that overexpress these herb proteins, and what you can see here is that comparatively to the cells, you see mostly the immature, uncleaved form. However, on the EVs, we see more of the cleaved, fusion-ready form of these herb proteins, and this was the case in the 3 herbs that I tested, but to varying degrees. So what seems to be happening is that there's a maturation gradient. The protein actually becomes mature as it's exported on the EV surface, similar to what would happen on a retrovirus itself. So now that we've shown that we can get these herbs onto the EVs, how can we actually track HERV-ENV positive EVs and their interaction with cells? So for this, we combined the overexpression of HERVs with POMGRET, which is a tracking molecule that allows for the inner leaflet of EV membrane to be labeled with GFP and nanoluciferase enzyme. And so this allows us to see where these EVs go, what cells they interact with, and so on. So we collected our EVs by ultrafiltration combined with size exclusion chromatography, and then after collection, we proceeded to characterize our EVs, of course looking for the HERV proteins present on the EVs, using transmission electron microscopy to assess the morphology. And lastly, we use nanoflow cytometry to look at the concentration, size, and GFP positivity, aka POMGRET positivity, of the EV populations. So we then moved on to measure our EV interaction with cells. So for this, we utilized 2 assays based on nanoluciferase enzymatic activity. So in the first one, which I call cell association, we measure all the EVs that are interacting with the cell, whether they're at the surface or whether they're internalized. We just simply wash away unbound EVs after incubating the EVs with the cells, and then we measure nanoluciferase activity. In our uptake assay, it's slightly different in that we remove any EVs that are at the surface of the cell by treating with trypsin prior to doing our nanoluciferase activity assay. So that can look only for EVs that are internalized. So we perform these assays with a set dose of EVs and various numbers of Hector NTPT or HeLa cells, incubating the EVs and cells together for 6 hours, and then measuring nanoluciferase. And what you can see here, and what I want to point out, is that specifically syncytin 1, which is one of these HERV proteins, enhanced our association and uptake with cells as compared to non-surface modified EVs that just contain the POMGRET molecule. Now, what's interesting is that the types of interactions depend very much on the target cell. For HEC293 T cells, this increase was only seen with association, but not with uptake. However, with HeLa cells, both uptake and association saw enhancement with syncytin 1 positive EVs. So we next wanted to test our EVs in more physiological situations. And for this, we specifically wanted to look at non-human primate PBMCs in blood ex vivo, following up on some of the work that Tom Driedonks did in our lab. So the reason we wanted to look at PBMCs initially is that if you're injecting EVs intravenously, PBMCs are going to be some of the first cells that the EVs will encounter. And if my goal in therapeutics is to get EVs to a location of the body, perhaps an organ or some peripheral site, a PBMC interaction would actually be a negative outcome. So we just wanted to see what would happen with these surface-modified EVs when we combined them with blood. So for this, we obtained blood from healthy juvenile male pigtailed macaques, and we mixed EVs with the blood for 30 minutes, and then isolated the PBMCs and performed flow cytometry to look for GFP positive cells. This is basically, essentially the association assay, but with GFP this time rather than nanoluciferase. Now, what we found, there's a lot of data here, but we tested this with 3 different separate animal donors, and interestingly, the primary cell that we see associated with EVs is B cells within the PBMC subsets. But there's also a significant amount of association with monocytes. But when we actually compare across different conditions, what was most surprising and interesting is that the EVs that have surface modification, whether it be syncytin 1, syncytin 2, HERV-K, or VSVG as a positive control, they all had reduced interaction with PBMCs in the blood as compared to the non-surface modified EVs that just contain the pongrat molecule. And when we break down the PBMCs by subsets, we see that this effect is primarily coming from the CD3 T cells, B cells, but I think most significantly the 2 types of monocytes that we measured, especially with these CD14 high monocytes. So the main takeaway here is that these surface-modified EVs are having what appear to be reduced interactions with PBMCs in ex vivo blood. So just to conclude here, what we found is that HERV envelope expression in cells leads to passive EV incorporation, and the EVs carry more of the mature, cleaved HERV envelope protein as compared to cells. EVs derived from XB293F cells that carry syncytin 1 on their surface have enhanced cellular interactions compared to control EVs, specifically with the target cells that we tested. Now, I don't know what would happen if we tested, we just don't know what other cell lines might happen because it's very dependent on the receptor identity, or at least that's what we think. But it's important to keep in mind that the target cell identity and indeed the producer cell identity might be very important to determining what kind of interactions take place between the EV and the target cell. And lastly, what I think was the most interesting finding was that the EVs carrying our HERV envelopes and VSVG had reduced interactions with non-human primate PBMCs in blood as compared to the control EVs. And this effect was primarily seen in the B cells and monocytes. So with that, I would just like to, again, thank the organizers for giving me this opportunity to talk today, and also to everyone in the Whitworth Lab and our various collaborators at Johns Hopkins and abroad, and of course my funding agency, Ionis Pharmaceuticals. And with that, I'm happy to take any questions. Thank you.
Hi, great talk. I just had a question. So I like the use of this luciferase, obviously, as a readout, but I wondered if you have any functional assays that you could test this in rather than using this biochemical readout.
Yes, that's a great point, and that's something we're currently working on, is developing the functional assays. So I'll get back to you on that sometime soon.
Okay, great talk. So when you look at PBMC association, I have 2 questions. I mean, in blood, PBMCs are, they're in a minority. The most abundant cell sources are erythrocytes and platelets. Did you ever actually look at the binding to these cellular components?
Yes, we did look at platelets. I didn't show the data here. It
see.
showed the same pattern of effects as the PBMCs.
And if you were to quantify the platelet binding versus all the other PBMCs, I assume you have much more binding to platelets than to the other cell components, right?
I'm not sure. I'm not sure of the quantity. So
I'm just thinking about surface area. You have
Yeah, I mean,
millions of platelets in a microliter of blood, whereas you have B cells are basically insignificant in comparison. And that's my own question. The other question is, why did you use non-human primate blood and not human blood?
because we have non-human primate animal models that we're planning to do in vivo studies with. So it made sense to me to initially test it with the non-human primate blood ex vivo. But of course we could test it with
Yeah, I'm just thinking, since you use human EVs, it could be good to also try human
I agree.
Thanks.
Thank you.
Very nice talk. I have one question concerning the types of the membrane fusion protein. There are many types of the natural and artificial the membrane fusion protein and peptide, for example, the HA2, the influenza virus, and GARA, the artificially the designed peptide, and so on. I'd like to ask what kind of the membrane fusion protein, the peptides, is suitable for EV cell uptake, for enhanced cytolytic release of the external contents, also the cellular uptake efficacy and so on.
Could you repeat the question? I'm not sure I understood.
Yeah, sorry. There are the many types of the membrane fusion protein, and I'd like to ask which kind of the membrane fusion proteins the suitable.
Which kinds are suitable?
Yeah. Have you ever tried to compare
Oh.
the kinds of these kinds of membrane?
No, I haven't. I haven't really compared to anything
Okay.
that's not viral or. Mostly, I've been working with the viral fusion proteins, like specifically VSVG or the endogenous retroviral ones. But I mean, it would be interesting at some point in the future to, do comparisons with other types of fusion proteins that exist.
Yeah. Each protein, the peptide has a different kind of mechanism, so
Right.
we need to compare. Thank you.
Fantastic talk. I just wanted to ask, do you know what the end organ distribution of these EVs with curves on them are?
That's something we're going to find out when we do the in vivo studies. Although we can at least try and predict what it might be based on what is known about the receptors for these ERV proteins and where those receptors are expressed. So I didn't show the table here, but there's, each HERV might have a different tropism to different organs based on receptor availability. But of course, until we actually test it, we don't know if that will be the case.
And what organs are the ones that are for the proteins that you've tested? Do you know which organs they target?
It's more of a broad tropism. Like it might be like the entire endothelial system, or in one case it might be liver, or I don't remember really specifics offhand, but yeah, that's not. I mean, we're gonna just try and perform these in vivo studies and see where the EVs go, and that'll be the data.
Okay, thanks.
Hi, kind of the opposite question. In terms of donor cells, do you know in vivo whether there's any particular cell types that express syncytin a lot?
Yeah, so the natural expression profile of syncytin 1 is the placenta, in the syncytial trophoblast cells of the placenta. So outside of that, its expression is generally restricted pretty heavily by methylation. So I have heard of it being expressed in cells in the testes and occasionally in muscle cells, but very low levels.
Let's thank our speaker one more time. So the next speaker is Mia Kordowski from Ohio State University, and she'll be talking about functionalized engineered EVs for targeted delivery to inner vertebral disc cells. Let's welcome the speaker.
Natalia Higuita Castro, and today I'm very excited to share some of my preliminary data on functionalized engineered extracellular vesicles for targeted delivery to intravertebral disc cells. So just to begin, to give an introduction on what the intravertebral disc is a joint between the vertebral bodies and your spinal column, and it absorbs shock and pressure during movement and allows your entire spinal column to rotate, flex, and extend. A healthy IVD should be strong, elastic, and flexible in order to resist compression. So there are two major regions of the intervertebral disc. There's the annulus fibrosus region, or the AF region, and this is comprised of type 1 collagen, and it's in these very organized concentric rings, and is comprised of fibrocartilage and AF cells as well. So this highly ordered type 1 collagen rings allow this AF region to have very high strength and stability and distribute loads across the entire intervertebral disc, and it also attaches to the vertebral bodies. And then the nucleus pulposus region is the interior region of the intervertebral disc, and it's comprised 70% water, so it's very gel-like in consistency, and it has also type 2 collagen and glycosaminoglycans, most importantly proteoglycans, which help retain the water content, and it's also comprised of NP cells. So this gel-like consistency of the NP allows it to absorb shock and diffuse nutrients. And the NP cells are also very important for synthesizing these extracellular matrix components, so the collagens and the glycosaminoglycans. So I like to refer to the intervertebral disc as like a jelly-filled donut. So the donut, the pastry part, is like the AF region that's more stiff, and then the nucleus pulposus is the jelly part, the inside. So in intervertebral disc degeneration, this is a prevalent cause of chronic low back pain, which is the leading cause of disability worldwide. So this can be caused by aging or injury to the disc or lifestyle choices such as smoking. And what happens specifically in the NP is there's a decreased production of the glycosaminoglycans and proteoglycans by these NP cells, which causes a decreased water content, which causes decreased cellularity of these NP cells, and it causes this NP region to lose its gel-like consistency and therefore not have that structural stability. And in addition, a degenerate AF will have, because there's less of those collagens being produced, it won't stay as organized, and so that mechanical stability will decrease. So overall in the disc, there's innervation, inflammation, and this can cause tears throughout the disc and loss of disc height, and this makes the disc more susceptible to herniation. So the current biological approaches to treat intervertebral disc degeneration have a lot of critical limitations. So cell-based therapies have been very difficult because of the unique environment of the intervertebral disc. It's a very low glucose, very low oxygen environment, and it has it's very avascular and aneural. So there's not a lot of flow of nutrients into this area. So direct cell replacements have been very difficult, and viral gene deliveries have been limited due to the recruitment of inflammation, which causes further degeneration of the disc. So our non-addictive strategy focuses on storing the structure and function and reduce the symptoms of pain. So in kind of this feedback loop I have on the right, there's this induction of degeneration, which causes damage to the environment, which causes degeneration of the IVD cells, which causes the cells to not produce the ECM, extracellular matrix components, and increase inflammation in the area, which causes this to continue degrading the environment. So we hope to intervene with EVs using transcription factors to deliver to the NP and AF cells and kind of hit the restart button on these cells to start producing these ECM components again. So we want to specifically deliver different transcription factors to each region of the intervertebral disc. So we have a Netrin-1 ligand that will specifically be uptaken by AF cells, and then a CD11A ligand that will be specifically uptaken by the NP cells. So I aim to use developmental transcription factors. So generally transcription factors, such as Yamanaka factors, will reprogram one cell type into another cell type. But again, the idea is to kind of reprogram from an unhealthy to a healthy phenotype, a pro-anabolic phenotype. So I'm going to first go through some already published data from the Higuita Castro lab to go over the transcription factors and the rationale and how they are able to bring these cells to a regenerative phenotype. So the use of FOXF1 and brachyury transcription factors for the NP cells, which is that jelly-like part of the IVD. So we have these NP cells in a 3D agarose hydrogel to mimic that gel-like environment of the intervertebral disc. And we injected EVs loaded with these transcription factors, FOXF1 and brachyury. We're able to see sustained expression after 2 and 8 weeks. And in terms of protein content of collagen and glycosaminoglycans, we're able to see increased protein content in the groups treated with EVs loaded with the transcription factors compared to sham, which is just the MC vector. And then we're also able to see age and sex-dependent gene level changes. So these are primary degenerate NP cells. So young female is going to be below the age of 60, and then older is going to be above the age of 60. And same for the males. So we're able to see a decrease in inflammatory markers, increase in anabolic markers, and decreases in apoptotic markers. And then looking at the reprogramming factors, transcription factors for the AF cells, are Moe-Hawkins boxes, or MKX and SCX for short. So this was a direct transfection experiment to test the efficacy of these transcription factors. We're able to see sustained expression, increased collagen protein content, and increased SOX9 expression, which is a marker of a healthy AF region, and a decrease in catabolic and biglycan markers. And I have the QR codes for all of these published works. And lastly, looking at the transcription factor FOXF1 for the NP region in a mouse model, we're able to see in histology and micro-CT images that the FOXF1 treated intervertebral discs in these mouse models that were directly injected into the IVDs had recovery similar to a non-injury compared to injury and a sham EV. And we're also able to see recovery of disc height. In addition, we did some behavioral testing on these mice. So in the blue is male mice, and in the red is female mice. And so the mice treated with FOXF1 loaded EVs had a similar regenerative effect as a non-injured invertebral disc. So this leads me to my part of the project, in which I'm hoping to specifically functionalize these EVs and be preferentially uptaken by the NP or AF and deliver those transcription factors. So we engineered donor human stem cells, and we do a transfection, and then I collected the EVs after 6, 12, and 24 hours, and I wanted to determine which had the highest gene expression and the highest concentrations and the most consistent size distributions. So from the gene expression, we're able to see that 6 hours seemed to be the most consistent, consistently high expressing, and the concentrations were highest at 6 hours as well, and the size distributions were most consistent at 6 hours. So then I wanted to confirm the presence of the ligand on the membrane of the EVs. So I wanted to use super resolution microscopy. So I used structured illumination microscopy, which takes 15 images of your sample at different diffraction patterns, and it overlays them and it reconstructs them. And by doing this, we're able to filter out the unfocused light and just have the focused light, so we can get a higher resolution image. So from this, we're able to see on the left we have the Netrin-1 loaded EVs, and on the right we have sham EVs. So the images in the middle are a zoomed-in image of the EVs at a 100 nanometer scale. So we're able to see for the Netrin-1 loaded EVs the red fluorescence of the ligands on the outside and the EV membrane on the inside. And then these were both stained with the same conditions. Lastly, I did a co-culture of degenerate NP and AF cells from primary autopsy samples, and we are still working on quantifying this, so this is just still preliminary data, but we're able to see that the CD11A functionalized EVs seem to be able to target the NP cells compared to just the transcription factor loaded EVs on the left. And same with our second group of the Netrin-1 being able to target the AF cells. So on the left are the transcription factor loaded EVs that seem to have no preference and are taken by both cell types. And then on the right are the Netrin-1 loaded EVs that are preferentially uptaken by the AF cells. So for future directions of this work, we want to deliver these EVs to the primary disc cells to determine if there's differences across ages, sexes, and disease severity. We also want to use the intervertebral disc mouse model that we used for the FOXF1 paper by puncturing these and seeing if there's a targeting efficacy in vivo as well. And then we also want to do proteomics on these two EV groups to see if there's differences in protein composition. And lastly, I just want to thank ICEF for inviting me to talk, and my lab, the Higuita Castro lab, and Geiger Perez lab, and then our collaborators, Dr. Divina Farmaser, Heather Powell, and Elizabeth Yu. And then I have the Higuita Castro lab website QR code and my advisor's information here as well. Thank you.
Thanks a lot for this nice presentation. I may have missed this point, but how did you check the loading efficiency of your different factors? Did you look at it or?
The loading efficiency. So we did PCR to confirm like gene expression, and so on different papers. So for example, for this FOXF1 paper. This one, we did like traditional PCR to confirm loading of plasmids as well into the EVs. And so that's how we kind of confirmed. And then in addition, I'm hoping to further confirm cargo loading using the super resolution for these EVs with ligands as well. Thank you.
Hi, really lovely talk. I'm just curious about your SIM imaging technique. Was this performed on fixed vesicles, or
yes.
It was? And
yes.
Is this something you can do on live to watch the biogenesis of the vesicles, or it can only be done on fixed?
Yes, we do. We are able to do it on live. I'm curious about doing that as well. We do have the ability to image it using SIM live. I think we can also do Storm live, the microscope that we have at Ohio State. But yeah, it's certainly something that would be really interesting to look at. Yeah.
Awesome, thank you.
Mm-hmm.
Very nice talk. I
Thank
have, I think, basic question. One question.
Okay.
I could not follow the how to express the lettering mark on EV membrane. So did you use, for example, the LAMP2B or other kind of the membrane, exosomal membrane protein?
So your question is, how do we get the natrin loaded onto the membrane?
Mm.
So what we do is we do a transfection of the donor skin cell, and we transfect it with the overexpression plasmid for the ligand, and then the cell packages the EVs, and we just collect them, and they have, it's a transmembrane protein, so it's in the transmembrane of EVs. So it's just engineered by the donor cell.
Okay. So have you ever tried to optimize the different kind of the membrane protein or EV membrane protein? Maybe the influence the expression level of the objective protein and so on. So this method is the best method.
Oh, I'm sorry. The question is.
The question is, I think there are many methods to express the in and the membrane of the EV. So I'd like to ask why there is your method.
Yeah, I'm definitely familiar with the other methods like coating it with like antibodies, the EVs to get ligands on the membrane. The reason that we moved forward with this approach is because in our lab previously we were able to show that we're able to load the ligands on the surface of the EVs just by doing a transfection from the donor cell. And because we're already transfecting with the transcription factors, we just continued with this method. But we do hope to do further characterization to really show that the ligand is on the surface and not loaded in the lumen of the EVs.
Thank you.
Thank you. Thank you.
Thank you.
So the next speaker of the session is Sarah Stewart from Los
how proteins cross membranes, and this is particularly in a mammalian cell sense. So obviously all of you are probably very aware of the conventional protein secretory pathway, where a signal peptide directs protein secretion into the ER, and it's then trafficked through the Golgi via secretory vesicles into the extracellular space. However, there's a subset of proteins that lack a signal peptide that are also secreted into the extracellular space. And so this is known as unconventional protein secretion, or I'll be referring to it as UPS. So there's a few different pathways that proteins can take to access the extracellular space. One of these is direct translocation across the plasma membrane. And this can be either facilitated or non-facilitated. So facilitated potentially by a transmembrane protein within the plasma membrane. Of course, there's also secretion via vesicular intermediates, and which needs no introduction here really, is extracellular vesicles is an example of this. But we also have autophagosomes and secretory lysosomes as well. So I'm particularly interested in a range of different proteins that are secreted by UPS, and this is just a table of some of the proteins that we're interested in. It's by no means an exhaustive list. So these proteins have both intracellular functions as well as well-defined extracellular functions. It's a very small percentage of the protein that's actually secreted, indicating this is a really regulated process. There's no common sequence or motif for secretion, like a signal peptide, and secretion is reported from many different cell lines. We know that it's not due to cell death. As I said, it's a very specific process. And interestingly, most of the proteins that we're interested in also are found both solubly in the extracellular space as well as encapsulated within or associated with extracellular vesicles. And so when we were looking at this list of proteins, we also noted that several of these proteins are reported to bind lipids. So that piqued our interest in lipids. So today I'll be particularly focusing on annexin-A2 as a model protein. So annexin-A2 is a calcium-dependent membrane-binding protein, and it binds specifically to phosphatidylserine and phosphatidylethanolamine. So this very, very busy picture shows you that annexin-A2 has lots of different functions within the cell. But importantly, it has really well-described functions outside the cell. So it's involved in thrombosis and viral entry, and we know, and I'm sure lots of you have seen annexin-A2 in EVs as well. So we're interested in the membrane and the fact that lots of these UPS proteins actually bind to the membrane and interact with it. So that led us to think, okay, so what are some of the key regulators of maintaining membrane homeostasis? And we wanted to look at lipid translocating membrane proteins. And so these are some examples of lipid translocating proteins, and this includes flipases, flopases, and scramblases. And so we were particularly interested in the phospholipid scramblase, TMEM16F. And so to cut a really long story short, we found that TMEM16F is involved in UPS. So TMEM16F is activated by calcium, and it flips phosphatidylserine from the inner leaflet of the membrane to the outer leaflet of the membrane. It also flips phosphatidylcholine from the outer leaflet to the inner leaflet. So it's interesting to note that most of the studies on this protein are looking at it during high calcium conditions, so by a release of intracellular calcium. But it's important to note that it does seem to have some functions at a basal level, so not under stimulatory conditions. So what we did next to investigate its role in UPS was to generate TMEM16F knockout cells. And here we're using HeLa as a cell line, just as a workhorse really. And so we knocked out TMEM16F, and we could show using an exon 5 Cy5 binding that we no longer, when we stimulate with iomycin, which increases intracellular calcium, we no longer see phosphatidylserine moving from the inner leaflet to the outer leaflet, so we don't get an exon 5 binding. And this is two different clones of the knockout. I'm struggling with this. So next we wanted to know whether or not TMEM16F is required for UPS. And so we looked at an XNA2 and A5. So an XNA2, as I said, it's present on the outer leaflet of the plasma membrane, and it's a calcium-dependent membrane-binding protein. So we took advantage of this property, and we released an XNA2 from the cell surface using EDTA. And this allowed us to see how much an XNA2 was extracellular compared to how much an XNA2 was actually intracellular. And we could see that when we knocked out TMEM16F, so these are the lysates here, and these are the supernatants here. When we knocked out TMEM16F, we could actually see that an XNA2 there was a severe reduction in an XNA2 on the plasma membrane, indicating that TMEM16F is required for its secretion. And so we have this model here showing that TMEM16F is important for an XNA2 UPS. So next, we know that, so we've also shown that an XNA2 is not only present on the cell surface, it's also present on the surface of extracellular vesicles. So next we wanted to know whether or not TMEM16F was important for the retention of an XNA2 at the surface of extracellular vesicles, and in fact, whether or not it's important for biogenesis and function of EVs. There's some data out there already proposing TMEM16F as a regulator of EV biogenesis and function. So it's been reported to be important for shedding microvesicles, but again, this is in response to stimuli. This is either damaged cells or cells that have been stimulated with high concentrations of calcium. There's another paper that shows that TMEM16F is actually involved in multivesicular body formation. And again, that was under stimulatory conditions upon TCR engagement. Interestingly, our proteomic data from several different cell lines shows that TMEM16F is actually present in EVs, and this is supported by multiple entries on Vesiclepedia. So little work has been done on TMEM16F under non-stimulatory conditions, and so that's what we aim to do, is look at TMEM16F at basal levels in EV biogenesis. Okay, so in terms of what we did and how we did this is we used the knockout cells, and what we wanted to do was essentially just collect all vesicular material that was secreted by the wild type and the knockout cells. So here we're not separating out any of our large and small EVs, and we're just using ultracentrifugation. So I know this is a bit of a, hammer kind of blunt force kind of approach, but it allows us to analyse what's out there in the supernatant. And so we could see that compared to the wild type EVs, we see a twofold decrease in both of our knockout clones of particles released per cell. We can see that the particles that are made by these cells still retain that cup-shaped morphology. And then, so we went on to characterise these EVs a little further. We can see that they're positive for CD81, CD63, CD9, and a whole range of EV markers. But interestingly, we could also see that these EVs had a reduction in an XNA2. So that suggests that TMEM16F is actually really important for not only either biogenesis or secretion, but also protein packaging into the EVs that are released. So next, we know that an XNA2 is impacted, are other proteins impacted. And so we did mass spectrometry on the wild type and knockout EVs, and we could see that there was a change in the protein profile, and we have differential expression of different proteins in these EVs, including a loss of TMEM16F itself in the knockout EVs. So this is all very well and good, but I suppose, coming to more of a functional assay, what's the impact of TMEM16F on these EVs? And so we had our wild type and HeLa knockout cells, we generated EVs from them, and then we popped it into a pancreatic cancer cell model. I'm not saying this is an in vivo model, it really is just an in vitro sort of readout. But what we could show was that when we put wild type EVs from the HeLa cells onto pancreas cells, we could increase their cell growth, and you can see that there's more colony formation when they're incubated with these EVs. However, when we incubated with the same concentration of knockout EVs, this was completely ablated. And so we found this was really interesting. So, of course, it could be that these EVs have lost a really important protein that promotes cell growth, or it could be that these EVs are actually functionally defective. And so in order to look at that, and this is very new data, we decided to do an EV uptake assay. So we overexpressed CD63 GFP, we isolated EVs, and we put them on the same recipient cells. And what we could show is that compared to the wild-type EVs, the knockout EVs were not able to associate and potentially be taken up by these cells as efficiently as the wild-type EVs. So finally, we do want to know why TMEM16F is important for EV biogenesis and function. And so we're currently trying to characterize different organelles within the secreting cell. So we're looking at the plasma membrane. TMEM16F is definitely at the plasma membrane. That's where it flips lipids and is thought to have most of its function. However, we're also looking at the early endosome, multivesicular body, and lysosome as well. And so just as a real snapshot, because we've only just sort of generated this data, we can show that LAMP1 and CD63 co-localization is actually increased in the knockouts compared to the wild types. And the average puncta, hopefully you can see, you can appreciate here, there are sort of these clusters of fluorescence. The average puncta is increased in both LAMP and CD63. So hopefully today I've shown you that TMEM16F is an important regulator of unconventional protein secretion, and that its loss reduces EV secretion, alters protein packaging into or onto extracellular vesicles, and impacts its uptake and function. So now we're looking to repeat this in other cell lines, and we've got similar data for this in both cancer and non-cancer cell lines. We're investigating the different morphologies, which I've already discussed, but we're also going to do this by EM. I'm really curious to see whether or not we've got less intraluminal vesicles within the multivesicular body, for example. And of course, we want to investigate the lipid composition of these to see whether or not that's perturbed and potentially mediating the effects that we see. And we'll also look at the proteins decorating the EVs. So I'd just like to thank everyone who's worked on this project. I have a small subgroup within Professor Suresh Mathivanan's bigger group, but thank you to everyone that's been involved and my previous mentor, Kevin Morrow.
Hi, thank you very much for this talk. I'm Xabi from Hungary, and I have perhaps a simplistic question, but it's intriguing to me that clearly you see a loss of EV secretion and function with this knockout. Did you think of doing the reverse? So if you overexpress it, will you get more secretion and more function?
That's a really good question and something we're working on currently. So yes, we do. We've got several plasmids that we can use. So we do have rescue cell lines, and we know that we can rescue the phenotype that we see with the soluble form of an XNA2. So we're just going to work on seeing whether or not we can rescue the EV phenotype that we see. But interestingly, we've also got a constitutively active form of TMEM16F. So I'm really curious to see whether or not it is that activity that's essential.
Well, that's very nice. I'm also curious. So thank you.
Hey Sarah, great talk. Natalie Turner. So I noticed at the end there you mentioned you might look at lipid composition. I always find now with omics studies, to me it's very one-sided if you just look at one omic. Like if you just look at a protein, you're kind of ignoring all the other, like what else is it interacting with? So are there any plans to do more thorough characterizations, not just lipids, but maybe transcriptomics and anything else that it could be interacting with in the cellular environment, either at the membrane or otherwise?
Yeah, it's a good point. At this stage, we're sort of only investigating lipids and proteins. I know that lots of people also work on RNA and things like that, but yeah, it's definitely something that we can think about doing a little bit more omics. I guess we're trying to take a fairly targeted approach, knowing that TMEM16F is responsible for maintaining lipid homeostasis. So, yeah.
If I can squeeze one question. I've got a really naive question. I'm really interested in the rate of flipping of some of these proteins, and I'd love to get your insights. Are there any methods, picosecond methods, that can actually measure them directly? and to see how that changes with lipid composition and genetic modification, so or is that an opportunity?
I suppose it's potentially an opportunity. I think we were looking into ways we could visualize this process, so this lipid flipping. I did put in a grant to try and work out the methodology.
That'll solve it.
To look at it. It didn't get up, so we're no closer. But yeah, no, it's a really, really good point. I think that actually visualizing this in real time would give us significant insights into the role of TMM16F, particularly in UPS. But even if we could visualize it during EV biogenesis, I think it would give us some really good insight.
Thank you. Let's thank our speaker one more time. So the last speaker of the session is Maria Harmadi from the Institute of Biochemistry, Hungarian Research Network, Biological Research Center. She'll be talking about quantitative features of EV-mediated crosstalk in multicellular 3D tumor models.
Good morning, everyone. First of all, I have to tell you that's a great honor for me to give a talk here at the ISAC, so I would like to thank the organizers for this opportunity. So as you heard, my talk is about the EV crosstalk in 3D tumor models. This is an early stage project. So tumor development is a complex evolutionary process where the tumor cells adapt to their ever-changing conditions, such as the selection pressure of the immune system, the hypoxia, or the therapies. And this adaptation highly relies on the interactions between the tumor cells and their micro and macro environment. Tumor cells are able to control the function of the non-malignant cells to work for the tumor's benefit, and they use complex signaling pathways for this purpose, like direct cell-cell contact, soluble factors such as growth factors, and they also use extracellular vesicles. You know that countless number of studies described the tumor's ability for the re-education of the stromal cells in the tumor microenvironment. And some papers also highlight the bidirectionality of this communication and the essential role of stroma, such as cancer-associated fibroblasts like extracellular vesicles. However, this communication is more complex because the stromal cells also communicate with each other, and although we have knowledge about the individual communication axis, but don't have any comprehensive picture how the network works, how the individual EV axes relate to each other. This figure represents the journey of EVs from the producing cell to the recipient one and highlights some key questions regarding the EV biology. And to answer these questions, some prominent researchers of the EV community stated that there is a need for multicellular 3D model systems for tracking the extracellular vesicles at a single cell level. Taking their advice, we created a multicellular 3D tumor model system for live tracking the extracellular vesicle crosstalk. We aim to identify the primary communication axis in different tumor types and drug treatments. So this means that we aim to study whether it is the control of the tumor cells which is prevalent in the tumor microenvironment, or whether it is, for example, the fibroblasts that overtake this function and control the OR network. It is also an interesting question whether this network setup varies from tumor to tumor. So we integrated biological, technical, and also computational developments into a single workflow. There are 4 key points of this workflow, namely the in-cell EV labeling, the application of 3D tumor models, the high-throughput imaging, and the machine learning-based image analysis. Let's see them in more detail. So first of all, we use the in-cell EV labeling to avoid the artificially overdosed EV concentrations. So instead of using exogenously administered extracellular vesicles, we aim to study the endogenous physiological EV crosstalk. So we stain the donor cells, including tumor, fibroblast, and endothelial cells, with different cell tracker dyes. These dyes are converted into fluorescent membrane impermeant products in the cytoplasm, so they are incorporated into the daughter cells during cell divisions, but their horizontal transfer can only be achieved by extracellular vesicles. In this way, the transfer of the vesicles can be easily followed in living cultures. After stainings, the cells are co-cultured in 2D or 3D plates and then subjected to high-throughput imaging. For the 3D experiments, we use a self-developed patented high-content screening plate, which was designed for live cell imaging of 3D structures using light sheet microscopy. After the imaging, we use machine learning-based software, the so-called bias software, to the quantitative analysis of the EV crosstalk at a single cell level. And finally, we use the RStudio for statistical analysis. Here you can see what these models look like. Until today, we have compared breast cancer, a melanoma, and an osteosarcoma model, and here are some representative images of their 2D cultures and 3D spheroids. And if you take a closer look into the 3D structures, you can observe red tumor-derived spots or gray fibroblast-derived spots in the green endothelial cells. But let's see the numbers behind the images. First, as an example, I would like to show you the 24-hour melanoma model. Values on this figure label the number of the detected EV spots for all of the 9 communication pathways. And as you can see, tumor cells and endothelial cells showed high self-EV transfer, while the crosstalk was the most active in the fibroblast endothelial cell and the tumor endothelial cell axis. Red numbers label the detected spots after a low-dose doxorubicin treatment, which was generally increased the EV communication. This slide shows the EV communication activity for each donor cell type in the different tumor models and culture conditions after a 48-hour incubation time. So the diagrams show the detected EV spots by the cells they originate from and the distribution of the vesicles in the different recipient cells. What you can see here is that most of the cells targeted the tumor cell type. For example, the tumor cells primarily targeted the tumor cells. And an interesting point that the fibroblasts, which are represented by the middle columns, transferred the least EVs in the 2D cultures, but they were more active in the 3D cultures. If we restrict the analysis to the crosstalk between the different cell types, you can observe that not the tumor cells, but the stromal cells were the most active. And they targeted each other. I mean that the fibroblasts primarily targeted the endothelial cells, and the endothelial cells primarily targeted the fibroblasts. As I mentioned, in the case of the melanoma model, the low-dose doxorubicin treatment increased the EV communication activity in most of the EV axes in the 2D cultures, but the drug affected mostly the endothelial cell and the tumor cell, between the endothelial and tumor cell crosstalk. In contrast, in the 3D cultures, where additional factors arise, such as drug penetration differences, the communication activity sometimes decreased instead of an increase. These results inspired us to further improve the model system, so we switched the spheroids into a model which mimics the extracellular matrix as well. We created hydrogel-based models using the TrueGel 3D technology, and this hydrogel ensures the adherence and also the migration of the cells but makes the single cell level analysis much more easier. And recently, we also take long-term images, and we added a fourth type of cell into the model system, the THP1 undifferentiated monocytes rebuilt with cell tracker Violet. Here you can see a comparison of the 2D hydrogel and spheroid model system. It's logical, but still surprising that the hydrogel was closer to the 2D cultures. And it highlights that we should be careful in designing such kind of model systems. Now let me summarize this short talk. From a technical point of view, we have created a multicellular 3D tumor model system for live tracking the EV crosstalk. Regarding the biological questions, current data suggests that stromal cells have primary role over the tumor cells in the EV crosstalk, but each of the tumor has a unique communication network. For example, in the melanoma, endothelial cells transfer the most EVs into the fibroblasts, and in the breast cancer model, the fibroblast cells transfer the most EVs into the endothelial cells. But what are the benefits of this project? For example, we aim to compare different tumor types and also subtypes to reveal the individual features of the communication strategy, of the tumor's communication strategy, and we also plan to isolate cells which internalize high number of extracellular vesicles using a modified patch-clamp system, and we want to perform molecular analysis of them. We will also do further quantitative data analysis of the EVs as well, and once all the tasks are done, this model system will be suitable to predict drug effects. In my opinion, this is very important because chemotherapy can work as a double-edged sword. It can kill the tumor cells, but it may also enhance the adaptation process of the tumor cells. And this adaptation is reflected by the EV-mediated communication. This model will be also appropriate to test EV blocking and might be suitable for developing high-throughput personalized medicine platform too. Finally, I would like to introduce you the team, without whose help this project would not be possible. I am especially thankful to my group leader, Kristina Buzás, and my colleague Ákos Diósdi for their contributions. Thank you for listening attentively. That was a great pleasure to share my ideas with you, and I'm looking forward to your questions and constructive criticism also here, or you can contact me via email.
Thank you for your talk. It seems that, if I understood correctly, most of your crosstalk or EV release is actually self-release.
Yeah.
So how relevant do you think EVs are for crosstalk in tumors?
Sorry, can you repeat?
So since most of the EVs that are released
I understood that.
Yeah, so how relevant do you think the EVs in crosstalk between the different cell types is actually for tumor development, rather than...
How relevant?
Yeah.
Because in the introduction, I tried to explain that the tumor cells are able to control the function of the stromal cells in the microenvironment, and this is very important, this process, and how the stromal cells reflect to this manipulation. And I think it is very important, this overall crosstalk in the fate of the tumor disease.
Hi, really, really nice talk and really beautiful imaging. I'm just curious, in your 3D models with your light sheet, if you've ever seen any dying cells in any of your tumor spheroids. Purely selfish question.
So, not understand exactly. So, did you ask that did we see dying cells.
Yeah, do you ever have any situations where you actually see a cell on the outside of your spheroid die and pull away?
Actually not. Maybe in the hydrogen model I saw some cells which explode. Yeah.
Okay, thank you.
Great talk. I also like the images a lot. So, if I understood correctly, the method by which to track the EVs was lipid dye, like cell tracker dyes?
Cell tracker dyes and cytoplasm dyes.
Cytoplasm dyes, okay. Well, I guess my question doesn't make sense then. Thank you.